Welcome, medical students, to today's microbiology lecture on the fascinating topic of bacterial genetics. In this comprehensive script, we will delve into the intricacies of genetic information within bacteria and its crucial significance in the field of medical microbiology. Bacterial genetics encompasses the study of heredity and variation in bacteria, providing insights into their cellular processes, evolution, and pathogenesis. Bacteria possess a single circular chromosome, which carries essential genetic information necessary for their survival and reproduction. These genetic traits can influence various disease outcomes, antibiotic resistance, and adaptation to diverse environments. Understanding the fundamentals of bacterial genetics forms the basis for numerous advancements in medical microbiology. The structure and organization of bacterial genomes play a crucial role in their genetic function. Bacterial genomes consist of a circular DNA molecule tightly packed within the nucleoid region of the cell. Several important elements are found within this structure, including coding regions, genes, non-coding regions, regulatory sequences, and mobile genetic elements. These components collectively determine bacterial characteristics, such as virulence factors, metabolic capabilities, and antibiotic resistance mechanisms. Plasmids are small, circular DNA molecules that can exist independently of the bacterial chromosome. They contain additional genetic information that confers advantages to the bacterium, such as antibiotic resistance genes or virulence factors. Plasmids can be classified into various types, including conjugative, non-conjugative, resistance, and virulence plasmids. Genetic exchange via plasmid transfer plays a pivotal role in the spread of antibiotic resistance among bacterial populations and contributes to the adaptability and survival of bacteria in different environments. DNA replication in bacteria is essential for cell division and inheritance of genetic information. It follows a semi-conservative mechanism, producing DNA molecules with one original and one newly synthesized strand. Enzymes like helicase, polymerase, and ligase participate in replication. This knowledge is crucial for developing antimicrobial treatments. Bacteria have evolved exquisite DNA repair mechanisms to counteract the constant threat of DNA damage. Exposure to genotoxic agents or errors during DNA replication can lead to mutations or genetic instability. Multiple repair pathways, including base excision repair, nucleotide excision repair, and homologous recombination, exist to preserve the integrity of the bacterial genome. Failure in these repair mechanisms can result in genetic mutations, genomic instability, and increased susceptibility to antibiotic-induced DNA damage. Bacterial transformation refers to the uptake and incorporation of naked DNA from the environment into bacterial cells. This process enables bacteria to acquire novel genetic material, including antibiotic resistance genes or virulence factors, through horizontal gene transfer. The transformation machinery involves specific proteins, such as competence factors and DNA-binding proteins, which facilitate the internalization of extracellular DNA. Understanding bacterial transformation helps elucidate the potential for genetic exchange and the spread of antibiotic resistance within microbial communities. Conjugation is a significant mechanism of genetic transfer in bacteria, primarily mediated by plasmids known as conjugative plasmids. It involves direct cell-to-cell contact, typically using a specialized structure called a sex pilus. The transfer of plasmids allows for the dissemination of genetic traits, such as antibiotic resistance or virulence determinants, between bacterial populations. Unraveling the intricate molecular details of conjugation aids in understanding the rapid spread of antibiotic resistance and pathogenicity in bacterial communities. Phages are viruses that infect bacteria. They replicate within the host and lyse the cell. Occasionally, bacterial DNA gets packaged into phage particles, resulting in transducing phages. These phages can transfer the bacterial DNA to other bacteria, facilitating genetic recombination. Transduction plays a crucial role in bacterial evolution and adaptation. Gene expression regulation is vital for bacteria to respond to changes in their environment, modulate bacterial growth and survival, and adapt to different host conditions. Bacteria employ various regulatory mechanisms, including transcriptional regulators, sigma factors, riboswitches, and small regulatory RNAs, to control gene expression. Understanding the intricate molecular details of gene regulation enables the development of targeted antimicrobial strategies and the unraveling of complex bacterial behaviors. Understanding bacterial mutations and mobile genetic elements is crucial for combating antibiotic resistance. Mutations occur due to replication errors or DNA damage. Mobile genetic elements, like transposons and integrins, promote gene transfer, increasing genetic diversity and dissemination of harmful traits. The rise of antibiotic resistance poses a significant threat to public health. Bacteria acquire antibiotic resistance through various mechanisms, including genetic mutations, acquisition of resistance plasmids, or horizontal gene transfer involving mobile genetic elements. Understanding the mechanisms by which bacteria acquire, maintain, and spread antibiotic resistance genes provides critical insights into the development of effective antimicrobial strategies and the prevention of resistance dissemination. Genomic islands are large segments of DNA that have been horizontally transferred into bacterial genomes and have significant impacts on their adaptation and evolution. These regions often contain clusters of genes that provide distinct advantages, such as antibiotic resistance or virulence factors. By studying the structure, acquisition, and functional implications of genomic islands, we gain valuable insights into bacterial pathogenicity, environmental adaptation, and their potential as targets for therapeutic interventions. Through understanding bacterial genetics, we can combat infections and develop antimicrobial strategies. DNA replication, gene transfer, and antibiotic resistance are critical in these processes, highlighting the significance of this field in medical microbiology.